Welcome to the Manitoba Ag Days podcast, where we hear from some of the most relevant, up-to-date, informational speakers in our industry. In episode 9, Richard Cuthbert, wheat breeder at Agriculture and Agri-Food Canada, shares his story. We have the wheat genome sequence. Now what?
Okay, thank you for the kind introduction. This is a pretty interesting venue to be giving a talk in. I'm living in Swift Current now, and I understand the Wheat Kings play here, and I don't know if the Broncos are really accepted here right now, but I grew up in Winnipeg, and I cheered for the Bombers for a long time, and I still cheer for them in Saskatchewan, and people like Curtis still give me a hard time about it. So the title of my talk is we have a wheat genome sequence, now what? And you could take that a couple ways. It could be now what or now what? And you kind of heard from Curtis some of the positive things about the wheat genome sequence and what we can do with it. And I'm going to put it into more of a practical term and some of the positives and challenges we still have with it. So I come from Swift Current, right in the heart of the the brown soil zone, typically the driest and hottest area of the prairies in Canada. And we also have a satellite site in Indian Head, Saskatchewan. There's 5 people that work with our group from Swift Current in testing new varieties and new genetics for release. And we're part of a broader system in AAFC. We work with groups all across the country. I'm just one kind of cog in the system. But we work with people from everywhere from the Peace and Beaver Lodge down to Lethbridge, where they have the winter wheat breeding programs, more CPS breeding, into Saskatoon, Melfort, down to Indian Head, Brandon right here. We have colleagues that we work with extensively, Santosh Kumar, Andrew Birch are the breeders here, and then into Morden and even further into Ottawa and PEI. We're testing material right across Canada. And that's just AFC. We're working with people like Curtis Posniak, of course, and other researchers to improve wheat. So just to look at wheat across Canada, what's been going on the last 5 years. We have a chart of insured wheat classed acreages, and you've probably taken a look at these from the Grain Commission. They've done a good job in putting together these summaries of what wheat's being grown, and it's pretty representative. It's only about 76% of the actual acreage, but it gives a good idea of what classes are being grown. So I simplified it into a chart, and on the top we can see overall acreages since 2003 to 2017 and an average over that time. And we can see it's kind of declined. We've kind of averaged out at about 17 million acres, and CWRS is by far the bulk of them, followed by Durham, followed by CPS Red, and then we kind of get into the winter wheats, hard whites, soft white springs, CNHR, and some others. Just a bit of a background on wheat. Curtis has given a very good introduction to what we're talking about in wheat and wheat breeding, but a few more points. Wheat, if you're not aware of it, is still 1/5 of all calories and protein consumed globally. So it's a very important piece of nourishment for the world. Canada is producing roughly 30 million metric tons annually, and that's a proportion of the 740 million metric tons that are produced globally annually, but generally quite high quality. The International Wheat Genome Sequencing Consortium, which Curtis Posniak just talked about, developed this infographic a year or 2 ago, and it's a nice way of looking at how bred wheat has come to be. So about 250,000 years ago, 2 species came together to form Triticum turgidum, and Triticum turgidum is actually the progenitor of durum wheat today. Triticum turgidum, about 8,000 to 10,000 years ago, hybridized with Aegilops toshii, and that's what gave us modern bred wheat, and that's what we've been working on improving over the last 8,000 to 10,000 years. Curtis just mentioned this, but the wheat genome is huge. It's a massive wheat genome. It's 17 billion base pairs. It's 5 times larger than the human genome, which Curtis said. And I don't know if Curtis said it, but it's 105,000 genes, roughly. That's a lot of genes that are involved in conferring that phenotype. What we see, what we harvest, what we eat. And improving bread wheat varieties, we want better, not more. We don't need more of the same. We need better yield, better agronomics, better disease and pest resistance, and better end-use quality. And this was actually 2 pictures from our demo site in Swift Current on irrigation, red fife this summer versus one of our newer lines, AAC Elida VB, a midge-tolerant wheat. And you can see there's some definite differences, shorter straw, much stronger straw, it's not laying down, there's not a big hole in the middle of the plot, and a much, much better yield, much, much better quality. So that's what we're after. And in the registration process, we can break it down into these 3 components: agronomics, disease and pest resistance, and end-use quality. That's really how the registration system looks at it to assess merit for registration. Curtis has mentioned some of these things, but I'll reiterate some of them because they're important. Agronomics. In a new variety, producers need varieties that are resilient. 2016 and 2017 were excellent examples of that. They couldn't have been much more diverse years. And we need varieties that do equally well between those two types of years and others which are unknown. So at the top, I've highlighted yield, grain yield and grain protein. And just on the right, I've taken a graph of our registration test in Western Canada this year, just to show that yield and protein are negatively correlated. So if you want to get more yield, just forget about the protein a little bit. If you want to get more protein, you kind of have to forget about the yield a little bit. But there are ways to shift that. So if we look at the lowest yielding line in the registration test, this is actually Katepwa, a variety from 1980. And we can see that we've generally shifted to the right on yield, and we've shifted kind of up in some of these for a better relationship of yield and protein. And that's how things like AAC Brandon have come to be, and the first speaker this afternoon, you saw Prosper and Brandon. That isn't a coincidence that the yield and protein are different. It's the genetics and how they were bred. After that, Curtis mentioned maturity. Well, it's easy to get higher yield if you forget about maturity, but we're kind of fixed with about 100 frost-free days in most of the prairies. You want the ideal height, strong straw strength, threshability, but you don't want to thresh too easily because it could shatter, and not too hard. Test weight, kernel weight, water use efficiency, or drought tolerance, nitrogen use efficiency. I don't think anybody here wants to put down more nitrogen than they need to achieve the desired yield and protein. And cold tolerance, I'm sure a lot of people want to get in the field as soon as possible, and then we get into great production. When we get into disease and pest resistance, there's a number. By far the most complex and the one we spend the most time on is FHB, Fusarium head blight, and mycotoxins, or vomitoxin accumulation in the grain. So it's bolded at the top. And then leaf rust, stem rust, and I also have stripe rust bolded. That's an evolving pathogen which Curtis was talking about, and we spend a lot of time incorporating new resistance for that. And then common bunt, loose smut, the leaf spot complex, which is 5 different diseases, ergot, and then tests, the wheat stem sawfly, which is important in southwest Saskatchewan, but not so much here. And then orange wheat blossom midge, which is becoming a problem in much of the prairies. And Curtis mentioned durable genetic resistance. We don't want resistances that are going to break down in a year after they're released or within 5 years. So we kind of try to incorporate multiple forms of resistance. Coupling adult plant resistance, which is usually quantitatively inherited. It's more of a scale from 0 to 10 versus on and off like a light switch. So we pyramid these types of durable genetic resistances. I'll just speak a little bit about each of the diseases in a little bit more detail, but Fusarium head blight is by far the most concerning across the prairies and what we spend our time on. The end result is Fusarium damaged kernels, and you can see some examples of different checks that we use in our breeding programs. So ELSA, which would be an MS, berry, which would be an intermediate, carberry, which would be a moderately resistant, 5602HR. So you can see the Fusarium damaged kernel variation between them. And unfortunately, mycotoxins are done. We can't see. We need a lab test, and that's expensive. Resistance is quantitatively inherited. I kind of spoke about a distribution. So on the bottom here, we can see a genetic population of related individuals at 2 different FHB nurseries. And you can see it's that bell curve, kind of a normal distribution. So from 0 to 100%, And you'll see very few are at the 0 to 10%, which is where we would want to be. There are some major loci, like FHB1, you may have heard of if somebody talked to you about wheat or you've looked into it, but none of these loci have been officially cloned. There's actually 3 or 4 groups in the world that say they've cloned it, and all of them have said that it's a different gene. And we know the genes, they've been sequenced, so this is an issue, and for things like gene editing, it's a real issue because we can't actually start to use gene editing without knowing the gene. The sources of resistance were originally quite diverse. They came from Asia, things with names like Sumai 3, Wuhan, Moringa, Wangshubai, and if you saw these in the field, you would not want to grow them. Unfortunately, we don't have to work with them much anymore in CWRS. Just a mention about Fusarium head blight ratings. Curtis touched on this as well, and our rating does not imply symptom-free. There is one spring wheat that I'm aware of that's registered in Canada that has an R rating. This is a picture of it in the Morden 2017 FHB nursery. And you can see, if you saw it in the field, you might not think it was actually totally resistant for its rating. There are hits, and you can see the characteristic whitening of the heads in certain places. The good thing about that type of resistance in tenacious is it stops and it won't really spread in the head, but it's not completely immune. So how we come up with those ratings is kind of complex. Pathologists do this. We call it the ISD, so it's incidence, severity, and dawn. So incidence is the number of plants affected. Severity is how badly those plants are affected. And then deoxynivalenol content in the grain. And it's weighted more to dawn, 60%, than it is to incidence and severity. And once this ISD is calculated, we can compare it against some known checks that are rated resistant to susceptible, and we can come up with that rating. It's not 100% perfect, but it gives a pretty good idea of where a new variety would sit. And these are just some examples of checks that we would use in the Fusarium nursery. You can see AC berry on the left, it's rated an intermediate. You can see hits throughout the row. The incidence is high, the severity isn't too bad. And then AC Elsa, the incidence and severity is bad, it is totally white. And then something like Carberry, there are periodic hits within the row that could spread over some time. The diseases, I've spoken about the rust, leaf rust, stripe rust, or yellow rust, and stem rust. We also work on common bunt, loose smut, the leaf spot complex, and even a little bit on ergot resistance. And the most concerning, as I mentioned, is yellow rust, as it's started to adapt to overwintering in Western Canada, and it's becoming a pretty major threat. We do have very good resistance out, but we're continually trying to incorporate new forms of resistance and pyramiding them. Wheat stem sawfly isn't much of a problem here, but you can see the larvae eating within the stem that was laid into it by the female laying the egg. Our main source of resistance is solid stem cultivars like Lillian or AAC Concord and CWRS. Orange wheat blossom midge is much more of a problem here. Our main mode of resistance is the SM1 gene. Any variety having that, of course, is subject to the midge-tolerant wheat program. You can see what happens when you have a midge-susceptible variety in a midge-infested field. You'll have that typical damaged kernels where the larvae is fed on the kernel, deteriorating quality. And then we get into end-use quality, and I'll speak mostly about Canada Western Red Spring today, but because it's the most restrictive class and because it is the most grown class of wheat in Western Canada. So it's used for the production of high-volume pan bread. It's used alone or it's mixed with other wheats to be used to make steam bread, noodles, flatbread, or common wheat pasta, hearth bread, and our export markets really quite like our CWRS because of its versatility and high protein content. And these are some of the things we need to be aware of in breeding those CWRS varieties. So we have pre-harvest sprouting. And on the right here, you'll see some heads, 10 heads from something that looks good and 10 heads from something that looks bad. I'll tell you that those are both registered varieties, and in the seed guide, if you see something with a sprouting rating of very poor or VP, that's what that is. It's not good. If you leave that in the field for in the fall and it's rained on, that's what it will look like. So we incorporate pre-harvest sprouting resistance or some dormancy into our varieties to try and keep them looking like this if they do have that weathering. We do this work by collecting heads in the fall in plots, specifically grown for this, and then they're put into a specialized growth cabinet where rainfall is simulated. So pre-harvest sprouting is important, and the falling number would be one of our main ways of looking at pre-harvest sprouting resistance or tolerance. Then we're interested in grain protein, of course, both in the flour and in the grain. And then we get into milling. So flour yield is very important. We want a high extraction from the kernel. That's dollars in the miller's pocket. And then flour ash and kernel texture. And then we get into the nitty-gritty of gluten strength and extensibility and how the dough is handled. And we have a few terms or equipment that we use for that, a mixograph, a farinograph, and extensograph. So this is a farinograph curve. This is an extensograph. So the farinograph mixes the dough up. The extensograph stretches it out. We want something that's strong and extensible. There is a joke with some of the scientists that you just take a word and you add a graph to it and you have a new quality measurement. And then we get into the litmus test, baking. So water absorption is important. The more water they can put in the dough, the bigger the loaf, the more loaves they can sell with the flour they put in. So bakers like that. Crumb structure is important. We want the nice white, tight crumb loaf and a high loaf volume, nice and fluffy. And then in some cases we get into noodle parameters as some of our export markers use our wheat for that. So just a quick word on wheat class modernization. I won't spend a lot of time, but we've gone through a consultation and implementation of changes to targets for gluten strength and CWRS, CPS, and adding a new class which has become CNHR. And I just want to talk about the range. Protein really hasn't changed. Protein's kind of stayed the same between them, but the gluten strength window has narrowed considerably. So this is where we were. This is where we are now, so it's tighter. If we were to use curling analogy, hitting CWRS before was like getting into the 12-foot, into the 8-foot. Now you better have a piece of the button if you're going to have any hope of hitting CWRS classification. So it's restrictive, and in every restriction we place on a breeding program, we already have FHB resistance we're trying to work on, and now this type of restriction, it's important for our end-use markets, but it's another restriction on the program which complicates what we do. There's a genealogy of CWRS. Our group has been putting this out for quite some time. It's due for an update. I think the last one that came out was 2012, and a lot's changed since then. So I just wanted to point out that you can look at things right from the original crosses in Canada. So hard red Calcutta and red Fife, which developed, I believe, is that Marquis? And you can see some things became backbones on the genealogy. So this is probably Niboa. And you can see how they became backbones. And they became backbones in CWRS because of their quality profile and always trying to get back to that CWRS quality. So our breeding strategy in Swift Current is to use low diversity in our parents to maintain our quality profile. And we try to minimize diversity because that typically brings in linkage drag on economically important traits. Like if we brought in Fusarium head blight resistance, we'll probably drag on CWRS or yield or protein. And we typically use low cross numbers, so about 24 three-way crosses and about 12 doubled haploid populations a year. Some programs would be up around 200 to 300 crosses a year. And we use larger populations. As Curtis was mentioning, the large populations are important to mine recombination. And we try to incorporate simultaneous qualitative and quantitative trait selection and this is important especially in Swift Current because we're working on heat and drought stress. And we don't exactly have other ways to select for them except for selecting for yield. So far as I'm concerned, the best predictor of yield is yield. So this is kind of an example of our breeding pipeline. So we work on about 24 three-way crosses a year. So we'll pick two parents and we'll cross them. We get an F1. And if we're in hybrid wheat, my job would be done and we'd all take off. But we're not yet. So we cross it to something wider. This is usually where we brought in some diversity, and then we top cross with something that smooths it out a little bit, and something like Brandon would be an ideal top cross parent here. And then we do a bulk increase, and we can even run markers at this stage if we've produced enough seed. And as Curtis said, for every marker we run, we have our population. So if we produced 1,000 seeds here, which is a lot of crossing because every kernel's a cross, if we produced 1,000 and we ran one marker, we'd be down to 500 seeds. If we ran two, 250. If we ran another one, 125 seeds. And we still want to get seed off that for selection, right? So then we put these into a space-planted disease nursery in Swift Current, and what does that look like? So I've got a drone shot here. So this is our F2 nursery, one of them in Swift Current. You're looking at roughly 300,000 genetically distinct plants in a disease nursery. So you can kind of see the irrigator hanging out here. So we inoculate with leaf rust and stem rust, the seeds inoculated with common bunt, and then we were able to select for plant type, the height, straw strength, and maturity. Unfortunately, you can't tell yield on a single plant. And then we get into maybe natural stripe rust, orange wheat blossom, midge. And out of those 300,000 plants, how many plants do you think become a variety on average? One. So you got to find the one. If we could find the one faster, we'd be good. But we usually end up picking about 20,000, 25,000 single plants. And those single plants, the seed from each one gets a one-way ticket to New Zealand or Chile. And that, I've called this real-world speed breeding. There's a lot of talk about doing 6 generations a year. We do 2, but we do it in the field, and we're able to actually select for important traits along the way. And the other thing, if you're only doing one plant at a time, you don't get enough seed to do yield plot, usually. So with these rows, one plant to one row, we get enough seed to do yield testing in the next year, and we can actually look at those rows for important traits like straw strength, plant height, and some diseases and maturity. The ones that get to come back go into yield trials at Swift Current and some other locations, and then we get into plant height, lodging, maturity, and the most importantly, grain yield and grain protein. And then test weight, thousand kernel weight, and then we get into quality on the selection, so kernel hardness, flour yield, gluten strength. And simultaneously, all these entries are in FHB nurseries. I see some people around here that help us out with those nursery. I think Roger Larios is sitting right there, and Carmen is waving. So these are very, very important nurseries. We rely on collaborators quite a bit, like the University of Manitoba and Carmen, Morden with Maria Antonia Enriquez, Brandon, the wheat breeding crew at AFC, Andrew Burt and Santosh Kumar, and we're starting one in Indian Head, Saskatchewan. And this is where we use a visual index to rate material and select for good and bad. Unfortunately, we can't do a lot of dawn because every selection would have to be threshed, ground, flour analyzed in a lab for dawn content, and it is quite expensive. So we do it strategically. So I just have a flyover. We can't actually go and look at one in person, but this will give you a pretty good idea of what it's like to be in an FHB nursery. So this is just a drone flying over top, and you can see the rows that are really bad, the really white ones, they're obviously thrown out. And you can see some material that's nice and green. Those are the ones we want. And we wouldn't have a clue what we're doing without these types of nurseries, so they're very, very important tools. And is it working? I think we're making progress. In CWRS, these are a few advanced lines, and there's a lot more, but these ones got me made me happy to see in the field. You can see something that's not looking very good beside them and looking quite green beside those. And then a couple weeks later, when other things have kind of round right off from FHB, these ones are still looking good. So the genetics are there. We need to understand them better, and we're working towards that to make the small improvements. DH, Curtis spoke about this. This is another way we can take high-value crosses, something like, let's say, AAC Brandon and AAC Viewfield or AAC Landmark, cross them, and we can develop segregating populations in 1 year, and we can even run markers on those when we're developing them. They'll be pure breeding in a year. The other way, we're probably taking upwards of 4 years. And a lot of the varieties you're growing came from this: Lillian, Carberry, Muchmore, Stetler, Connery, Viewfield, Redberry, Elida. Wheat breeding efficiency. It's ultimately efficient use of resources, so the tools, the yield plots, the nurseries, the quality testing, the double haploids, remembering that 300,000 F2 plants, about one's a variety. And if we make about 8,000 DHs a year, about one plant's a variety. We need to find them faster. but we're bound by recombination and meiosis. Population size is important in this. We want to increase our gametes in what we're looking at to increase recombination. It's a random process. And assessing progeny with precision and accuracy is imperative. Discard the worst as quickly as possible. We had a cytogeneticist in Ag Canada that used to say that was the best tool a breeder had. We actually have a technician in our program that says 99.9% of what I do is garbage, but it only takes one. So we can improve what we're doing, and Curtis has kind of talked about this, using markers. So we can, a gene that's tagged with a marker, we can select for it. So in real terms, this is 1,600 plots. Let's say $50 a plot at 3 locations, $240,000. If we didn't discard the lines before that included an economically important gene that was detrimental, half of them are garbage. So we spent $120,000, and this is unreplicated for nothing. But if you actively use that marker to discard, all 1,600 are useful, and you'd end up with a better variety because you have more to look at for improved yield, FHB resistance, and quality. And the cost to screen with the marker, I would say in this day and age, $2 a line is probably pretty reasonable, so $3,200. It makes a lot of sense. But what if you could look at all the genotypes or genes of the lines and plants in early generations of the breeding program? And we're doing this in humans today. You can spit in a tube and you can send it away, and they'll tell you that you're 30% South American, 40% European. They'll tell you have a 56% chance of getting type 2 diabetes. And they're doing this through trait association to markers. So we want to do the same thing in wheat, and Curtis talked about this with genomic selection. So hopefully this isn't a copyright infringement, but it's 23andMe, and now it's 42 in Triticum, right? So the wheat breeding chip, cost-effective genotyping, it all hinges on how cheap we can get that. So we've been working on that with Curtis, and I think we're pretty much in a place where that's becoming a reality. So all this data we've generated in the program every year of the 99% that's garbage, it's useful, especially if we can have the genotypes that are behind it. So we can take the yield and the FHB and the quality data, and we can develop fancy statistical models once we've genotyped, right? We have the phenotype, we have the genotype together, genotype it, develop the marker, the model, and we can figure out what markers are important, and we can apply those to the 300,000 F2s in a perfect world, and we would be able to make better selections and make better use of our resources. So I just wanted to say quickly, I'm running out of time here, but AAC, in the last year there's been a lot of talk about drought resistance or drought performance, and this is the Western breadwheat C, the checks from it, in 2017 standardized against an old variety, Katepwa. So we've kept that in from the 80s until today. So we're able to look at new varieties against old varieties, and these letters mean significantly different. We can see that view fields out-yielded Katepwa over 11 sites, averaged 21%. And there's been some thinking that short varieties have short roots, poor drought performance, but that's obviously not true here. Now these are two fields, the same field, same population, randomized twice. One's rain-fed and one's irrigated. They're about 150 feet apart. Same block of land, same day, a drone shot on either one. So this is some of the drought stress work we're doing at Swift Current, and it's to understand the drought-related traits, so yield, stability, roots, leaf waxiness, and quality. And with the new genomic tools, like the wheat genome sequence and all these new markers we have, we're combining that together to improve our selection process to better understand why things like view field do that much better. We have some pretty good ideas, but they need to be proven. So where are we going? It looks pretty similar to Curtis's, I would say. Access to tools increase efficiencies and leads to better varieties. Genomic selection will allow the bad to be discarded quickly. Gene editing is a promising technology. We need to understand the genes involved. And phenotyping remains king. If we don't know the phenotype, we don't know much else. So a lot of our work is funded through producer checkoff, and we're very grateful to that. So it's a sincere thank you to the producers and the commissions that fund our work. I hope that you're seeing the benefits of our breeding in your fields, and hope we can continue to do that for you. So thank you.
We have time for one quick question, if anybody has a question for Richard. Are you around, Richard, a little bit?
A little bit,
if you
yeah.
need to be? Yeah.
Yeah.
So up at the top?
Sure.
Okay, so if
Perfect.
anybody has any questions of Richard, he'll be at the top. Fred wants to
Okay.
ask a question.
Yeah, I noticed the great presentations, and I understand disease resistance, but in Manitoba, we've developed an excess moisture issue. Is there any work being done on that?
Do you want to read?
Well, I would say Curtis probably not in Durham, but in high yield. I'm speaking for him, but in Durham, the FHB issue is pretty significant. So I think most of their work in excess water environments would be fusarium head blight resistance breeding. For me, it would be similar. We do test our advanced material across the prairies to look at what performs better. We're typically into more yield stability of what will do well in southern Manitoba and swift current. So you're selecting for the optimal stability. Not to say low yield, but optimal. For super excess water, no. I did my schooling at the University of Manitoba in canola breeding, and I know exactly what you're talking about. Days when you didn't know if you get the cedar into the field and things, but not specifically, no, I wouldn't say so. Curtis, did you? Excess water screening for variety development.
So please join me in thanking Richard for coming out today.
Thanks for tuning in to the Manitoba Ag Days podcast. We'll see you next year from January 22nd to 24th, 2019.